As Shamin just mentioned, today's topic will be emerging viruses. So within the next 1 to 2 hours, we will learn about the origins of emerging viruses and the determinants of their spread, and we will have a look at a few specific examples. So essentially, I could also have used the title outbreak for this talk, because it will also be about pandemics, epidemic outbreaks, and we will see how viral and environmental factors determine the spread of viruses in the human population. So I've brought a few pictures here as a teaser. So these are a few things that we will talk about today. So some of these things have obvious connections to viruses, but some of them may not be that obvious. So we will see later on what, I don't know, stacks of tires or tattooing, for example, has to do with viruses or emerging viral diseases. When you think of emerging viruses, you may, for example, think of the COVID pandemic of SARS-CoV-2, but maybe some science fiction movies may also come to your mind that refer to or were about outbreaks and viral diseases. But today we will learn what the true science between or behind these emerging viruses really is. So that's the outline for today. So first we have to define what emerging viruses are, and we will see how relevant they are, and we will see where emerging viruses usually come from. So what's their origins, where and how do they appear? And as I mentioned before, we will learn which factors really determine the successful spread or unsuccessful spread of a virus in the human population. And in the second half of my presentation, I will provide a few more examples or talk about a few more examples in more detail. And we will also talk about different scenarios of outbreaks. So does every outbreak result in a pandemic, or are there different scenarios that are possible? I wanted to make this also a bit interactive. So I have a few QR codes where you can answer questions virtually. We'll just see how this goes. If there's any question, then also just feel free to interrupt me or to put something in the chat if that's possible. So don't be shy. And I actually wanted to start with a virtual question. So if you have your smartphone ready and the QR code scanner, you can just scan this one. And I actually wanted to ask you, why, in your opinion, do you think is it important to study emerging viral diseases? So why is this relevant to us, to the human population? What reasons come to your mind? So if you scan this code, or if you type this address here, you will find a small text box, and you can just write down your answer. I give you 1 or 2 minutes, and then we can just collect a few ideas and see. I hope this works. If not, let me know. Does it work? Maybe Shamin can let me know.
Yes, Daniel, it's working.
Okay, perfect. Okay, yes, this looks good. Thanks so much. So I can see a few answers already. You can also see them. So someone put public health. That's good to prepare for future outbreaks. That's a very good point. So essentially, it tells us that we have to be prepared, right? It's not just that we need to react to emerging viruses that are already there, but we kind of have to prepare. And if there's an outbreak, we have to react quickly. Yeah, so it's both the prediction and the reaction to outbreaks. Yes, so all of them are very similar. So someone put animal health here. That's also an important point because essentially it's both. It's of course human health that we focus on, but it's also animal health, and both are actually very closely connected to each other. Let me just refresh to see. Oh, yeah, that's very good. In silico models is very nice. So we will also have a few or have a look at a few in silico tools later on. So we can use them to predict, to model outbreaks. What else? Yes, to develop vaccines. That would also be an important preventive measure. Yes, but essentially, so it's all about public health and prediction, prevention, and yeah, restriction of outbreaks. Very good. Thanks so much. Okay, so what I brought here is actually a video from a newspaper or an online newspaper in Turkey, I think. And it's about an outbreak of a virus. And we'll just have a look at this short video. I hope the sound works. And then we can discuss how scientifically correct this actually is and how dangerous this outbreak or this potential new virus is. So let's just have a look.
Is a new pandemic originating from China expected to impact the world? A 37-year-old man is in critical condition after being attacked by monkeys in a Hong Kong country park and has tested positive for B virus, making him the first person in the city to contract the deadly infection. The B virus, transmitted by monkeys, had previously killed a veterinarian and a surgeon in China. The virus can remain on damp surfaces for hours. Like the coronavirus, the B virus initially gives flu-like symptoms that may progress to infection of the central nervous system. There is no vaccine against the virus.
Okay, so this was just a very short video. So if you're a layperson and you watch this video, you may be kind of concerned because they talk about the new virus called the B virus that obviously emerged in China, that infects monkeys, can be transmitted to humans, and it has killed a few people already. They compare it to SARS-CoV-2. They say that there's no vaccine to this new deadly virus. So as I said, as a layperson, you may be concerned about this. So what's this B virus actually? I'm not sure whether anyone of you has heard of this virus before. It is actually a herpes virus. So the official name would be herpes B virus or herpes virus simiae because it naturally infects different monkeys. And this virus has been known for quite some time. So it's widely circulating in monkeys that are kept in zoos, but also in wild monkeys. It can also be found in cell cultures, in labs that use cells from monkeys. So that means everyone who is exposed to monkeys is at risk, including animal keepers or tourists, for example, that also get in contact with monkeys. And usually this herpes virus doesn't cause severe disease in monkeys. So it just causes like lip blisters, like herpes simplex virus infections usually do in humans. But if the virus crosses the species barrier and is transmitted from monkeys to humans, then it may actually cause a very severe disease and encephalitis in humans that is lethal in most of the cases. So some of the information in this video was correct. It is a deadly virus. It can cause lethal infections in humans, and there is no vaccine. But I don't think it's a new threat for a pandemic because so far there's no evidence for human-to-human transmission, and herpes viruses are DNA viruses, so they don't adapt, they don't mutate that quickly. So I don't think you have to be concerned about this. So this is a virus that has been circulating in monkeys for quite some time, and I wouldn't say that this poses a public health threat. So I just wanted to use this as an example of how there might be some misinformation or misleading information in newspapers or online web pages that may be difficult to interpret for laypersons. So to get some more scientific or better information, you could, for example, have a look at the webpage of the World Health Organization that is called the Disease Outbreak News, or DONs. So this is shown here, so we can just have a look at this together. So I just put the link here. So this is how it looks like. Disease outbreak news, and when you scroll down, you can actually see different outbreaks listed by date, and you can see the latest report is actually from November 5th. It's about an outbreak of Rift Valley fever virus in Senegal and Mauritania. And if you go further down, you can see there's outbreaks of chikungunya, Nipah virus, Ebola virus, and so on. Whenever you click on one of these, you get more information. So the WHO summarizes the specific outbreak, talk about the number of infections, the number of deaths, the possible routes of transmission, and so on, and also measures to contain the outbreak. So that's a very good webpage that I can really recommend, and we will have actually more detailed look at a few specific examples in the second part of my presentation. So as I mentioned initially, we have to define the term emerging viral diseases or re-emerging viral diseases. And the WHO says that emerging viral diseases are viral infections whose incidence has increased in recent decades and or will increase in the near future. So that's pretty trivial maybe, but what's important to know is that we can consider or refer to more than 10% of all human pathogens as emerging pathogens. And that does not only include viruses, but also other pathogens. What I've put here at the bottom is a timeline of emerging viruses, and you can see it's just random examples from the last few decades or years, starting from HIV in the 1980s up to the COVID outbreak in 2019. And what you have to keep in mind is that emerging viruses is nothing that is unique to the present, but there have always been emerging viruses also centuries ago, and there will probably always be emerging viruses in the future. And this is what I would like to indicate with this word here. So just keep in mind that emerging viruses may appear anytime. And not only any time, but also anywhere. So there are outbreaks of epidemics, of pandemics or endemics everywhere, all over the world. So independent of the climate or the population density, they can appear anywhere around the globe. And although we talk about viruses today, just keep in mind that there's different types of pathogens that can cause disease, and essentially most of them have the ability of causing emerging diseases. So apart from viruses, there are fungal infections, bacterial infections, parasites, and prion diseases that all of which can cause disease and may newly emerge. And one prime example would be prion diseases, which had not been described a few decades ago, let's say. But today, as I mentioned, the focus will be on viruses. So apart from any time and anywhere, it could also be anything that causes an emerging infectious disease. So just to go back to our overview, so now we have defined what emerging viruses are, and we have seen, or you have already known this, that they are very relevant for both human health, but also animal health. But now let's talk about this point. So where do they come from, and what determines, or which factors influence their spread in the population? So let's go back to this timeline here. So if you have a look at all these examples, so it's mostly viruses, and if you think about the origin, so I'm not talking about the geographic origin, but the origin of the virus. So where does the virus come from? How did it actually enter the human population in all these cases? So what do these viruses have in common regarding their origin? And the answer is that all of them are actually zoonotic viruses. So that means they have their origin in an animal host. And this is actually true for the vast majority of all emerging viral infections. So more than 75% are zoonotic viruses. That means they have crossed the barrier, the species barrier from animals to humans. And I would like to introduce a few terms here. So the species, the animal species where the virus naturally replicates, so the natural host of an animal virus is often referred to as the reservoir species. It could be rodent, but it could also be any other species, a primate, for example, like as we have seen for the herpes B virus. And what you can keep in mind is that the virus very frequently does not cause disease or only mild symptoms in its natural host in the reservoir species, potentially because it has co-evolved with its host for thousands or even millions of years. And then the virus can be transmitted from this reservoir species to humans. This can appear via different routes, either directly or indirectly via vector species. We will talk about this in more detail later, or it can be indirect. So let's say there's a wild animal like a rodent or monkey where humans usually don't get in touch with. So, but humans may have more contact to pets or poultry, for example. So these species may serve as so-called carrier species. So in this example here, the cat may catch the virus from a rodent, a mouse, and then the cat transmits the virus further on to humans. And there is actually a nice, oh sorry, this is just to illustrate again that the virus may not cause disease in its natural host, but it may potentially cause severe disease in humans. And there's actually a nice scene in this movie called Contagion that illustrates this transmission from a natural reservoir species to humans via a carrier species. So that's of course a science fiction movie, but a lot of the details, the scientific details in this movie are correct. And in this example, they actually show a bat virus in the movie that is transmitted to humans via pigs. So bats will be the natural reservoir and pigs are the carrier species. And we'll just have a look at this scene of the movie that actually illustrates this very nicely. So it starts with deforestation of the natural habitat of bats. So these fruit bats carry the virus, this hypothetical virus in the movie. They feed on bananas and they contaminate these fruits. And then these contaminated fruits can transmit the virus to pigs, for example, which serve as carriers. Okay, so in that movie, so there was actually the final scene of the movie, and the lady in the restaurant was patient zero, so the first human that was infected by this animal virus. Although this is science fiction, this might be a potential route of cross-species transmission. So humans don't get frequently in direct contact to bats, but they may catch the virus from the pigs. And I would also use this to introduce a few more terms. So we talked about the term zoonosis, but there are also these two terms, zooanthroponosis or anthropozoonosis, so that actually describe the direction of transmission. So if the virus is transmitted from the animal to humans, we refer to this as a zooanthroponosis. But actually, you have to keep in mind that human viruses can also spill over to animals. So if you're infected with the virus, you can transmit this virus to your pet, for example, and then we refer to this process as anthropozoonosis. Or if you just want to indicate that it goes both ways, you can talk about amphixenosis or diplozoonosis. These terms are not so common, but just in case you read about them, it might be good to know. Then there are also a few terms that describe the route of transmission. So I just told you that there can be a direct transmission from animals to humans or an indirect one. So if it's a direct zoonosis, then the virus is transmitted without a vector, or let's say without an animal vector like a mosquito, but via direct contact. So for example, if you pet your dog, or if you are bitten by your dog, that would be a direct transmission of the virus. Then parasites actually also frequently need a change of host. So some parasites have an intermediate host where certain stages of their replication cycle or propagation cycle occur, and a final host where the rest of the replication cycle occurs. We don't usually have this for viruses, so this term cyclozoonosis usually refers to multicellular parasites such as helminths. So that's also illustrated here with this number 2. Then there's something called metazoonosis. So here the intermediate host would be an invertebrate. So it could be a mosquito vector or a tick, for example. Then in contrast to zoonosis, there's also this term sopronosis, which means that the reservoir is actually not an animal species, but like the inanimate environment. So it could be a bacteria, for example, that is usually growing in soil and then transmitted from there to humans. And then one special type of zoonotic transmission would be a xenozoonosis, which refers to the transmission of a virus via xenotransplantation. So let's say animal organs such as heart valves from pigs are transplanted to humans. So if these animal organs are contaminated with the virus, then it may be transmitted to humans via surgery, via this medical procedure. Then, apart from zoonotic transmissions, viruses may also emerge via mutations. So as you know, viruses, especially RNA viruses, have a very high mutation rate. So this is illustrated here. So the highest mutation rates can be found among RNA viruses. It's a bit lower for DNA viruses and even lower for eukaryotes. But Still, viruses may accumulate lots of mutations over time, and this may in the end also result in the development or emergence of new virus species. And apart from single point mutations, also larger mutations or rearrangements in viral genomes may occur. And we refer to these as recombination or reassortment. And these two processes require the infection of a single cell with two closely related viruses. So this is illustrated here. So let's say you are infected with two viruses at the same time, this blue virus and the yellow virus, and they're closely related but not identical. In this case, they actually also harbor segmented genomes. That means their genomic or their genes is distributed on different segments. And if one cell is infected with both viruses, then the genome may either recombine or reassort. So recombination is shown on the right, refers to the process of genetic exchange within one genome segment. So this is illustrated here. You can see that most of the genome segments are still blue, so entirely derived from the blue virus. But there's one example of a segment where actually the genome segment of the yellow virus recombined with that of the blue virus, generating this chimeric sequence. And the term reassortment, in contrast, refers to the exchange or replacement of an entire segment. So there's no breakpoint in a segment where a new chimeric segment is formed. It's just a different assembly or packaging of different segments. And you can imagine that these processes, recombination and reassortment, in contrast to point mutations, have a much larger impact on the viral viruses that are newly produced and on viral evolution. In the end, however, all these processes may help viruses to adapt to new species. Let's say an animal virus crosses the barrier from animals to humans, then it has to adapt to its new host. So it has to adapt to an immune system that it has never encountered before. And these processes may help the virus to achieve this. These processes may also change the transmission rate, infectivity, potentially pathogenicity, or also help the virus to develop resistance against drugs or evasion from vaccine responses, for example. Another important determinant of virus spread is, of course, healthcare. This is pretty obvious, trivial. So let's say there are poor healthcare systems in certain areas of the world, certain countries, because of poverty, because of natural disasters. This will, of course, make it easier for viruses to spread in the human population. It can also mean that, for example, if we use antivirals in an inappropriate way, or if we use antibiotics in an inappropriate way to treat bacterial infections, we may trigger the emergence of drug-resistant pathogens. Another important determinant that is closely related to poor healthcare would be lack of immunity. And lack of immunity can refer to two different things. It can be the lack of vaccinations, so that in certain areas people are not vaccinated properly and do not have a good protection against, let's say, poliovirus, for example. But it can also refer to lack of natural immunity. So, for example, today there are also is quite a number of isolated populations, isolated peoples in areas of South America, for example, that usually don't get in contact with other people. So that means they don't get in contact with a lot of viruses that are circulating elsewhere in the world. So they don't have any natural immunity. And if a virus enters this population from outside, or several viruses, this may have detrimental effects. Then lack of immunity is similar but different from immunodeficiency. So lack of immunity can be the result of vaccination gaps, but immunodeficiency can have a lot of different reasons, origins. So for example, malnutrition or HIV infections can result in immunodeficiency. Also, increased aging or age is associated with immunodeficiency, and this is important, particularly today where people get older and older. So, with this, people may also suffer more or be more vulnerable to infections that do not cause disease in immunocompetent individuals, but only in immunodeficient individuals, and we refer to these infections as opportunistic infections. And one term that I would like to introduce in this context is the term syndemic. I mean, all of you know epidemic, pandemic, but syndemic describes the simultaneous occurrence of two or more diseases that negatively impact each other, so that benefit from each other, that may not cause severe disease if they occur separately, but severe disease if they could co-occur and infect you simultaneously. Then another key factor that determines spread of viruses in humans is, of course, globalization. So whenever we travel around the world, we may also, or the viruses may travel with us. Then, as you have seen in this example of the movie, they showed this example of deforestation, which means that humans get in contact to animal species and their viruses that they have never encountered before. And we also have to keep in mind that it's not only us and the viruses that travel via air traffic, but it can also be the vectors or carrier species. Let's say if rodents spread around the world, or if specific mosquito vectors spread, this may also facilitate spread of the respective viruses. And closely linked to this is climate change or global warming, because due to climate change, different vector species or carrier species, reservoir species may also reach geographic areas where they have not occurred before. And this is nicely illustrated by this map. So this map actually shows the current distribution of malaria mosquitoes in yellow, and the expected spread or distribution of this vector species by the year 2050. And you can see that it's predicted to spread to regions of the US, for example, but also Turkey or Central Asia, Australia, suggesting that in the future, not only the malaria vector, but also potentially malaria itself may occur in these areas. Another term that I would like to introduce, apart from zoonosis, is geonosis. So geo refers to the soil, earth. So that's more like a hypothetical scenario where a pathogen that, let's say, has been circulating hundreds of years ago survives in permafrost, for example. So let's say there was an influenza virus circulating decades, hundreds of years ago in Northern Asia, in Siberia, and it may still persist in this permafrost, let's say in burial sites, and with global warming, these burial sites or these viruses may thaw, may defrost, and they may still be infectious, hypothetically. So this has not happened yet, so there's no example of an emerging virus from this permafrost soil, but hypothetically this is possible. Then there are two terms that we need to talk about. This would be bioerror and bioterror. So bioerror actually means the unintentional release of pathogens. So we can refer to this as a lab accident. That's why we need specific safety measures. So we know these 4 biosafety levels that are used around the world, starting from biosafety level 1 to biosafety level 4, which would be the highest one, just to prevent or to enable researchers to work with dangerous viruses under unsafe conditions that prevent an unintentional spread of the virus into the environment. On the other hand, there's bioterror, which would be the intentional release of pathogens. And what you have to keep in mind is that these are not necessarily viruses that infect humans, but a bioterroristic virus could also be a virus that affects crops, for example, and could harm humans indirectly by destroying crops. Then another interesting example of let's say, newly emerging virus or newly emerging pathogen would be an infectious agent that has been known for quite some time, but was initially suspected to be harmless. And one example would be the bacteria Helicobacter pylori. So when this has been described decades ago, everyone thought that this would be a normal commensal in your gut, in your stomach, but then it turned out that it can actually contribute to disease and cause gastric ulcer and ultimately cancer. And something like this may essentially also be true for some viruses. So there are a few viruses that we know that we think are not pathogenic, but maybe, who knows, in the future, when research progresses, we figure out that they can cause disease or contribute to disease in certain situations. Or we have talked about prions before. Prions are a nice example for the discovery of an entirely novel group of pathogens. I mean, I've shown you these examples of bacteria, parasites, fungal infections, and viruses, but who knows if there's yet another entirely new group of pathogens to be discovered that is different from everything that we know today. So just keep that in mind also, that there might be yet something else to be discovered. And then with point number 14, this is my last point regarding the origins and causes of emerging viral diseases. I call this special risks or special practices. This refers to a lot of different things that may actually also contribute to efficient spread of viruses in the human populations. And this can include, for example, practices such as tattooing, special cosmetic treatments that involve injections that could pose a risk or facilitate transmission of viruses. In the case of tattooing would be hepatitis C, for example. Then I also put this image of the tire stacks here. These are important because actually rainwater accumulates in these tires, and they are excellent breeding grounds for mosquitoes that then can harbor viruses and serve as vectors. So there are actually a few countries and cities that do not allow stacks of tires for these reasons, just as a preventive measure. Then we briefly talked about xenotransplantations, which would also be a special practice or risk procedure that could enable the transmission of an animal virus to humans. And also certain ritual acts or religious procedures. So like decades ago, consumption of human meat was actually practiced in some peoples in Papua New Guinea, which allowed the transmission of certain prion diseases, or contact to corpses. Some embalming procedures may facilitate the spread of Ebola viruses in certain regions. So as I said, lots of different things, but just keep this in mind when thinking about factors that facilitate the spread of newly emerging viruses. With this, on this slide, I've just put together all the 14 points that we just discussed. So with this, I tried to classify or determine or define important determinants of viral spread. And what I would like to ask you to do now is just to think about a few examples of viruses and try to assign them to these different categories. So here I essentially just listed random viruses. So some of them may be well familiar to you, others may be not so familiar, but try to assign some of these viruses to some of these categories. So just think about which of these viruses have a zoonotic origin, which of them have an animal reservoir, which of these may benefit from climate change, let's say, because they are transmitted via a vector. You can discuss this with your neighbors. I would just give you, I don't know, a few minutes, and then we can try to collect your ideas and put everything together. Okay? Sorry, I will just switch screens because we can also make this in a more interactive way. So you should be able to see this, I think. So here on the left, there is again the viruses, the examples that I have. And in the center, I do have a few post-its. So yeah, just think about this. How would you assign different viruses to different categories? And then we will actually try to put them on these post-its together. You can also think about other viruses that are not part of the list. That's also fine.
Guys, could you please increase the font size for the list? It is not clear to the participant at the back.
Okay,
Yes.
sorry.
But now we cannot see the whole list.
I know, I'm sorry. But maybe, so should I switch to,
Now it's right. It's
pardon
perfect
me?
now.
Okay. Let's just try to do this together. I know that only one of you, I guess only Charmin has a microphone, right? It might be a bit difficult, so maybe Charmin has to answer all my questions now. But let's start with the first category, with the zoonosis. So which of these viruses do you know or do you think have a zoonotic origin, an animal origin? Maybe you can just talk to
I will
Charmin.
answer on their behalf. I will answer on their behalf. Yes.
Okay,
For the zoonosis.
yes, for the zoonosis.
Okay, one answer is Zika.
Zika, that's good. Yes. So
Ebola.
yes, what's the natural host of Ebola? Does anyone know? The reservoir?
Buffalo.
No, it's not buffaloes. It would be it's primates, but primates would be an accidental host. The natural host are most likely fruit bats, actually. Yeah. I heard HIV. Very good. A natural host of HIV would be it's also primates. So it crossed the barrier from gorillas and chimps to humans. And Ebola would be bats. Zika can also be
There's
primate.
also
Can you read this still?
SARS. And there is SARS also.
Yes.
Influenza.
Yes. Many species, including birds, for example. Excellent. Anything else?
West Nile virus.
West Nile, very good. What would be reservoir of West Nile? Birds, excellent. Anything else?
Chikungunya.
Yes, chikungunya, but it's not chicken. It's called chikungunya, but it has nothing to do with chicken. I'm just checking the list. MERS. Does anyone know what MERS is?
Marburg virus.
Marburg is good, yes. Marburg would be a filovirus closely related to Ebola. And Marburg also has its origin in bats. And MERS most likely is a coronavirus. It's short for Middle East Respiratory Syndrome virus. And it can be found in bats. And the carrier species are most likely camels, or are camels.
Hanta virus.
Okay, too many ideas. I'm running out of space. Hunter, very good. What's the natural host of Hunter viruses?
Rodents.
Yes.
What about yellow fever? There's also an option of yellow fever.
Yes, also true. In also primates. Nipah. Yes. What's the host of Nipah? Does anyone know?
No.
Could also be
No.
bats. And the intermediate host or carrier would be pigs. So essentially the video that I showed you from Contagion, so the movie is inspired by Nipah virus. So you just remember, so there was the bats that dropped the fruit that transmitted the virus to pigs, which then transmitted the virus to humans. That would be a true scenario actually for Nipah viruses, yes. Perfect. Let's leave it there for the first one. That's good. So this is also nicely illustrating that the vast majority is actually coming from animals and a lot of different animal species. So let's just zoom out a bit. Maybe we jump to climate change. So which of these viruses would you say benefit from climate change because they are transmitted by vectors such as mosquitoes that also spread more efficiently because of climate change? So which of these are vector-borne diseases? Sorry?
Dengue.
Yes.
The dengue virus.
Perfect. Yes, I heard yellow fever.
West Nile virus.
Mhm.
And okay, then again the chikungunya.
Yes. Yes.
Zika.
Perfect. I think that's looks good. I think that's the ones in the list.
West Nile India.
Okay. Perfect. Sounds good. Let's go. So it gets more difficult now. Let's talk about, let me zoom out for a second. Let's talk about reassortment and recombination. Which of these viruses can reassort or recombine or have recombined and reassorted? Any idea?
Influenza. The first answer the influenza.
For which one? Recombination or reassortment?
Okay.
Let's go for
Recombination.
okay. So influenza was good. That's the prime example for reassortment because influenza viruses have a segmented genome and they can reassort their genome sequences. Perfect. Yes. And recombination, So are you aware of any viruses that are circulating in humans that have recombined, where actually one genomic sequence is a chimeric sequence? Sorry, Shamin?
First option is HIV.
Yes, very good.
SARS, COVID-2.
Yes. SARS is most likely also the result of recombination and has also recombined during the pandemic.
Influenza here as well.
Probably yes. I'm not entirely sure, but most likely yes. Essentially, most of them or many of them can recombine and have recombined, but these are the prime examples. That's good. Yes.
MERS.
MERS. I'm not entirely sure, but most likely also. I guess so. Yes. That's good. Which of them, let's jump to immunodeficiency. Which of them would benefit from a host or a human with poor immunodeficiency? I mean, all of them would, but which of these viruses does not usually cause disease in immunocompetent in healthy individuals, but only in immunocompromised individuals?
HIV.
I mean, yes, HIV causes immunodeficiency, but I'm more asking for which virus would benefit from the immunodeficiency. So which of the viruses in the list would cause severe disease in immunocompromised individuals? Yes, but dengue can also cause severe disease in immunocompetent individuals. So there's one virus in the list that benefits from immunodeficiency. That is also especially apparent in AIDS, like in HIV-infected individuals. I think I heard it.
AIDS. AIDS with not the virus. Hepatitis.
Yes and no. I mean, hepatitis viruses can also cause disease in healthy individuals. So
then it's HHV.
yes, it's HHV8 because HHV8 is a herpes virus that usually does not cause severe disease, but it's also called KSHV, which is short for Kaposi sarcoma herpes virus. So it causes Kaposi sarcoma in immunocompromised individuals. So it's like in AIDS patients, for example.
Measles.
Measles, yes and no. So measles is an interesting example. Measles cause immunodeficiency, and would facilitate infection with other viruses. And essentially, all of these viruses would also benefit from some kind of immunodeficiency. But what I wanted to illustrate with the example of HHV8 is that a virus that usually does not cause any disease in healthy individuals may cause severe disease in some immunocompromised host. So I think we just leave it there for now with HHV8. That should be fine. Yes. So let's jump to lack of immunity. So I said that this can refer to vaccination gaps, for example. So where do you think, for which viruses is there a risk for vaccination gaps? So where do you think are may be not enough vaccination?
Polio.
Yes, that's a prime example.
Smallpox.
Yes, that's an interesting. I heard measles. Very good. Also.
Yes. Yellow fever. Yellow fever.
Yes, I think that's the best one. So polio, smallpox, measles. So smallpox have been eradicated, but they're still kept in two laboratories in Russia and the US for research reasons. So if they re-emerge, then the population may not really be protected anymore. I think in most countries, vaccination campaigns ended at the end of the 70s. And polio, so we are trying to eradicate them, but we haven't reached this goal yet. So there's still polio viruses circulating in some countries. What else? Let's jump to bioerror and bioterror. So which of these viruses would you think would be a risk for bioterror?
SARS again.
I wouldn't say SARS. I mean, you're saying that SARS may be the result of a bioterror, or that it could be used in future.
I would say.
For error, yeah, that might be true. I mean, there's still
They are saying bioterror.
yes, there's still discussion whether this may have come from a lab, whether this escaped from a lab in China. Yeah, that's a good example. I guess that's the best two examples that I would also have picked.
Zika for the bioterror?
Zika?
Yes.
I'm not sure whether Zika would be let's say a good agent. Sounds strange, but Zika depends on specific vectors, right? So without that vector, it cannot be transmitted from humans to humans, so it would not circulate. And so it depends on the country, of course. I think smallpox is the best example here, and SARS-CoV-2 is a good example for bioerror. There are also a few examples of unintentional release of influenza virus from labs. So most likely there has been a lab leak in Russia at some point that caused some outbreak of a specific influenza virus.
Or error or terror? Bioerror, the measles.
Yes, could be possible. I'm not sure. I don't think this has occurred yet, but would be bad, of course. Yes. Okay, sorry, I'm zooming out, so it's good. You have so many ideas, but what next? What about globalization? Which of these do you think benefit a lot because of human travel, migration?
SARS-CoV-2 globalization.
Yes, that's a good example. Dengue is also a good example. So I'm just hearing a few words, not everything. Dengue is good. So it's a prototypic example of travelers, at least for example in Europe, West Nile, similar. Essentially, it's most of them. HIV is a good example also. So many of them will of course benefit from human travel. Yes, also the Ebola outbreaks that have occurred in Africa, some of them were also transmitted to different or transported to different countries. Essentially, it's a lot, and most of them. Yeah. Yes. Then we talked about this category geonosis. So remember, this was the example with the permafrost. Some viruses that may be frozen somewhere in Siberia or somewhere else. Any idea what we could put here? Like some older viruses that may re-emerge?
Zoonosis.
Yes.
Any idea for zoonosis?
Smallpox.
Smallpox.
Good example, yes.
What else? Anything else?
Influenza.
Yes,
Influenza.
that's also a very good example.
HIV.
HIV probably not, because HIV is not that old yet. I mean, in principle, yes, but HIV just emerged roughly 40 years ago. But smallpox would be interesting because it has been circulating for quite some time. Influenza viruses also, so some older strains may reemerge. So let's leave it there. I know that this is not complete for sure, this list, so there's many more examples, but it is just to illustrate again these different categories. So what's missing? Special risks. So do you have anything in mind? Any weird procedures, risk behaviors that would promote the spread of any of these viruses?
SARS, HIV, influenza. Anything
what
else?
So what would be the behavior, the weird risk behavior for SARS-CoV-2, for Ebola, HIV, for the ones that you mentioned? Can you also specify the behavior or the risk?
Specify the risk. What are the risk factors? Name a specific risk. This around. Dengue, and what will be the risk? Climate change. So it will go to climate change, not to the. Okay. Dengue. The answer for the risk is the water, like what will I say?
Yes, it would be
Water storage or something. Yes. Then Amna, what did you say? Ebola. Risk factor? Yes. West Nile and Ebola and yellow fever. And what will be the risk factor? Direct contact. Okay.
Yes, these are good examples. I'm just thinking about something else. Yeah, probably you'll find many more examples for these different viruses, but I think that's good. Yes. For this one here, unknown etiology, that's a bit difficult because for all of these examples here, we of course know that they cause disease. So prime example here would again be the prions. Of course, these are not viruses, but they are unusual, and for quite some time they have not been known as a category of pathogens. So we can put this here. Or I had this example of Helicobacter pylori, again, not a virus, but it's a bacterium that has long been thought to be apathogenic, but is now known to be able to cause disease. And then I think this looks already pretty good. So the only thing missing was the mutations. Oh yeah, and pool healthcare. For mutations, we can essentially also put most of the viruses here because all of them mutate. The RNA viruses mutate faster than the DNA viruses, but all of them evolve. So that would fit to many viruses. And pool healthcare essentially also. Yeah, I would just put many here. But if we zoom out, so this is just to illustrate two things. So first of all, many emerging viruses have a zoonotic origin, and you nicely mentioned some of the reservoir species, in some cases also the carrier species. And this is also to illustrate that several categories may be true for a lot of different viruses, and that different viruses or single virus may also fall into different categories. So one virus may benefit from lots of different factors, essentially. Any questions related to this?
Questions? Until here? No? No, Daniel, you can continue. Daniel, you can
Okay.
continue.
Yes. Okay. Thanks. So my question would be, do we need a break actually, or should I just continue?
10 minutes break, Badlo.
Okay, then let's do a 10 minutes break.
Yes.
Is that fine? Okay, then we meet at, I don't know, it's for you, it would be 3:15, right?
3:07. Yes, we will be back at 3:15.
Okay, perfect. See you.
See you.
I wanted to pick a few very specific examples and discuss them in a bit more detail, and also show you a few tools that might be helpful for you.
Excuse
Sorry.
me, Daniel.
Pardon me?
Yes. Your screen is blurred. We can see only the blur.
You can only
Is it from
see.
your side or from our side?
I guess it might be your side. I can see it, but I can stop and share again. Let's try. I just try again. Better?
Yes, perfect.
Okay.
Yes, perfect.
Okay, good.
You
So one thing or one important thing that you have to keep in mind is the species barrier when it comes to zoonotic transmission. So that means we are not susceptible to all animal viruses because our immune system, our organism, of course, differs from an animal organism. And because of the coevolution of viruses with their hosts, there are certain so-called species barriers. And that's actually indicated here on the left. So the host is just indicated by this yellow antibody-like thing, and the virus by this green shape. And just keep in mind that the virus continuously evolves evasion mutations that allows it to evade the immune system, but the host may adapt to these evasion mutations and again reacquire the ability to target the virus. But in a different host species, this may occur in a completely different way. So another virus may evolve completely other different evasion mutations. So the host has to adapt also with different mutations. And because of this arms race and this co-evolution, viruses and their hosts, especially factors involved in antiviral immunity, evolve very, very quickly and evolve in different ways in different species. And because of this, there's usually a strong species barrier. So that means a virus that efficiently infects host A will not directly be able to efficiently infect and replicate in host B. So it has to adapt to its new host in order to spread efficiently. And this can be achieved by single point mutations or by larger genetic rearrangements as we have seen for recombination or reassortment. What you may also keep in mind here is that some viruses may be pre-adapted to the human host, especially if their natural host is a species that is closely related to humans. So let's say a virus that is circulating in chimps, in chimpanzees. For this virus, it may be easier to jump to humans because chimpanzees are our closest relatives. And the virus that infects cats, for example, may have a larger barrier to overcome because cats are much different from us. And because of the species barrier, there are actually also different scenarios of the fate or the ultimate fate of a cross-species transmission. So let's just go through a few examples. So let's say there's a reservoir species and a virus is transmitted from this reservoir to humans. And what actually happens in most of the cases is that the human host is resistant to this infection. And I've just put one random example here, which is FIV. So this is a virus that is related to HIV, the human immunodeficiency virus, and FIV is the feline immunodeficiency virus that can be found in different feline species, including the house cat, but also pumas, for example. And our cells are naturally resistant to this virus. So an FIV that infected your pet cat cannot infect your cells. So there will be no infection even if you're exposed to the virus. And this actually happens in the majority of cases. In some cases, there might be a so-called dead-end infection. So that means the animal virus is efficiently transmitted to a human individual and infects this human individual, but this human does not spread the virus further in the human population. And one prototypical example here would be rabies viruses. So let's say someone is bitten by a rabid dog, then the rabies virus might be transmitted. But because of the route of transmission, this infected human individual will not pass the virus on to other humans. So, and this person would be a dead-end host, so no risk for the entire human population. In some cases, however, the virus may further spread in humans after the infection of patient 0 via an animal host. For example, if the virus was already pre-adapted to humans, or if that particular human had a kind of immune deficiency that allowed the virus to replicate to low levels and to adapt. But even if the virus spreads in the human population, there are different scenarios. So there could be an endemic spread, like for dengue virus, for example. That means in some areas of the world that are geographically restricted, the virus circulates over time, and it's not limited. This outbreak is not limited in time, so it's restricted in geographically, but not in time. In contrast to this, the term epidemic describes an outbreak that is also limited in time, and the prime examples would be Ebola outbreaks or Marburg virus outbreaks that occur from time to time, but that can usually be restricted or eliminated within a few weeks or months without affecting, let's say, the entire human population. So these usually don't spread around the globe, so they are, again, geographically limited. And in only a very minor fraction of the cases, such a spillover event will result in a pandemic. SARS-CoV-2 is an obvious example where one single cross-species transmission most likely resulted in spread all over the globe, and HIV would be another example. But again, this is probably the most infrequent or the rarest case, a pandemic. In the vast majority of cases, we will not even be infected by an animal virus we are exposed to. Just keep this in mind. Then one term I would like to introduce is the term R0 or R0. So this is the so-called reproduction rate. And the reproduction rate is an important number for epidemiologists because it describes the average number of individuals that one people would newly infect. And this is illustrated here by this cartoon. So for Ebola virus, for example, the reproductive rate is rather low. So one individual, indicated here in black, on average infects only 1 to 4 other individuals. In contrast, so we've talked about measles already. Measles has a very, very high reproductive rate, infecting about 15 other individuals if there is no vaccination, of course. And obviously, a virus can only spread efficiently or continuously in the human population if this rate is above 1. If it's lower than 1, at some point, the virus will go extinct. And this reproduction rate, or this R0, R0 number, is actually also nicely explained in the movie Contagion. So we'll just have a look at one scene from this movie again, where a scientist, actually describes and explains this R0 number.
What we need to determine is this. For every person who gets sick, how many other people are they likely to infect? So, for seasonal flu, that's usually about 1. Smallpox, on the other hand, it's over 3. Now, before we had a vaccine, polio spread at a rate between 4 and 6. Now, we call that number the R0. R stands for the reproductive rate of the virus.
Any ideas what that might be for this?
How fast it multiplies depends on a variety of factors. The incubation period, how long a person is contagious. Sometimes people can be contagious without even having symptoms. We need to know that too. And we need to know how big the population of people susceptible to the virus might be.
So far, that appears to be everyone with hands.
Yeah, so she nicely explained this. So the numbers she provided are actually correct. And she said that this reproductive rate depends on a number of factors. So what percentage of the population is susceptible? Do some individuals have a natural immunity, acquired immunity? But also it would depend on the incubation period and the time someone is infectious without having symptoms. So if you feel healthy, but if you're already infectious, then the risk of transmitting the virus is, of course, higher. And this number is an important determinant to estimate the risk or spread behavior of a virus in the population. Then let's have a closer look at a few zoonoses again. So what I did is I actually selected a few examples, current examples, mostly from this year, from the Disease Outbreak News webpage of the WHO. And I picked Nipah virus. So obviously in the first half of this year, there have already been 4 fatal cases of Nipah virus infections in Bangladesh. And as you can see here below, it says the first 3 cases had a history of consuming raw palm sap. However, the 4th case had no history of consuming raw palm sap. So somehow there's a role of palm sap in Nipah virus transmission. We'll come to this on the following slide. Just briefly, what is Nipah virus? Nipah virus belongs to the mononega viralis. So it has a negative strand, single-stranded RNA genome. Actually, measles virus belongs to the same group, for example. So what's the role of palm sap? So we already mentioned or talked about Nipah virus being like the template for the movie, and that Nipah virus circulates in bats and can transmit the virus to humans via pigs. And one source could be the contaminated fruit, like the bananas shown in the movie. But another route of transmission would be contaminated palm sap. So obviously in some regions of India and Bangladesh and other countries, palm sap is harvested by putting these small bowls on the trunk of the tree. But unfortunately, bats also like this palm sap a lot, so they may drink from this palm sap, but then contaminate it. And then if humans harvest it, they may also ingest the contaminated sap, get infected, and be a patient 0, or then also continue spreading the virus in the human population. Another example that you already also put into one of our categories, or the zoonotic category, is Marburg virus. There has been a Marburg virus outbreak at the beginning of this year in Tanzania, which was declared ended in March, I think. Yes, and what they usually do is they wait for 2 incubation periods to see whether there's any new infections. And if there are no new infections during a double incubation period, then they declare, or the WHO declares an outbreak as ended. However, they also nicely write here that the risk of reemergence of Marburg virus remains after the end of the outbreak, linked to the animal reservoir's persistence or presence in the country. So obviously, even if you manage to eliminate a virus from the human population, if it's a zoonotic virus, may continuously spill over and cause new outbreaks. And this is different from viruses such as polio or measles or smallpox, which don't have an animal reservoir, because in these cases, we may actually entirely eradicate the virus if we manage to eradicate it in the human population via vaccination, for example. Then there's no risk of spillovers anymore from an animal reservoir, because there's just no animal reservoir for these viruses. However, for zoonotic viruses such as Marburg virus, this won't be possible. And obviously it's not feasible to vaccinate or to eradicate the virus from the reservoir or carrier species. Another closely linked or related example, another filovirus would be Ebola virus. There's an outbreak currently ongoing with several suspected cases, including deaths. And there are regular Ebola outbreaks in different countries of Africa, most of them being linked to some animal contact. So what you see here, I know it's very difficult to read, but this is actually a transmission chain described in a paper that tried to figure out the origin of the largest Ebola virus outbreak that occurred actually about 10 years ago in 2013 and 2014 in different West African countries. So each line here is a different individual that has been infected, and the arrows indicate who was, the transmission chain, so who was initially infected and transmitted the virus. And they actually could trace this large outbreak back to one index patient, so the patient 0, actually a 2-year-old child in a rural area. And this transmitted the virus, this child transmitted the virus to family members, and then at some point, a healthcare worker in a larger city got infected, and this healthcare worker actually transmitted the virus to other patients or healthy individuals and served as a so-called super spreader. So super spreader is someone infecting more individuals than usual, and unfortunately these are usually or frequently healthcare providers, medical staff, because they are the first that get in contact with these newly emerging viruses and that also see other patients and may transmit the virus, unfortunately, to additional people. Then we already talked a bit about recombination, and one of you mentioned HIV as a nice example for a virus where recombination occurs. And this is also illustrated here. The advantage or the reason why HIV tends to recombine is because it has 2 copies of a single-stranded RNA genome, and HIV is very diverse. So there's HIV-1 and HIV-2, so these are 2 different species, and HIV-1 can be subgrouped into different groups called M, N, O, and P, and M is the main group that can be further subclassified in different so-called subtypes or clades. So if one individual is infected with different clades or groups, these clades can recombine and form so-called chimeras or recombinant forms. And this is illustrated down here. So this is one example of a chimeric recombinant virus. So each box is one of the typical retroviral genes, and you can see that different parts of these genomes are derived from different parental viruses. So the parental groups of this virus are clades A and E, and what you see here is a circulating recombinant form that was derived from these 2 clades. And some of these newly formed recombinant forms actually have a selection advantage and may spread more efficiently in the human population than the respective parental virus. And that's also illustrated by this pie chart here, which shows the global or local distribution of individual subtypes of HIV. And you can see in pink that there's the circulating recombinant form derived from clades A and E, which is actually very successful in some parts of the world, for example, in Southern Asia, in India. So how can we determine whether a virus is a recombinant form and derived from 2 different other viruses? Oh, sorry, I forgot about this. There's also a nice movie scene in the movie Contagion where they actually talk about recombination. So the virus that causes the outbreak in the movie is actually also recombinant virus. So let's just have a quick look at this scene also. You have to pay close attention because it's just very briefly mentioned, but they say that the virus in the movie is actually recombinant of a pig virus and a bat virus.
We've sequenced the virus and determined its origin, and we've modeled the way it enters the cells of the lung and the brain. And the virus contains both bat and pig sequences. In the bottom right, you can see the dark green is pig and the light green is bat. And here you can see the crossover event, bat, and pig, bat. And here is a model of the virus and how it attaches to its host. The blue is virus, and the gold is human, and the red is the viral attachment protein, and the green is its receptor in the human cells. These receptors are found in the cells of both the respiratory tract and the central nervous system, and the virus attaches to the cell like a key slipping into a lock.
Okay, so before they talked about the receptor binding and how they solved this crystal structure, they showed this so-called recombination analysis. So she talked about breakpoints. That's actually the site in the genome where the camera was formed. And we can also easily do this online in silico. So in case of HIV, there's a nice tool that allows you to do so. So similar tools are available for other viruses or generally for different viruses. For HIV, this tool is called RIP, Recombination Identification Program. Let's just have a look at this, how this works. So this is the web page. I try to make this a bit larger. So what you can do is, so let's say you have isolated a virus sequence from a patient and you would like to test whether it's a known subtype of HIV or whether it might be a new chimera. And what you can do is you can paste your sequence here in this box. So in this case, we would just use a random sample input. So this would be a sample sequence, the entire genome of an HIV isolate. And now the software will perform a so-called sliding window analysis. So it will essentially cut the sequence into different fragments, and for each fragment, it will align this fragment to known clades to check whether it's derived from clade A, B, C, and so on. And this may identify breakpoints in the genome. So let's just do this. So down here, we can ask the software to perform an alignment of our query sequence with known subtypes. So subtypes A to L. And so there's different options here, but we won't go into detail here. So let's just select submit, and then it may take a few seconds. And what we get is something like this. So this is a so-called similarity plot. So let me just explain what you see. So down here, I can also try to make this a bit larger. So on the x-axis, you can see essentially the position in the viral genome from the 5' end to the 3' end. And on the y-axis, you see the similarity to specific reference sequences. So essentially, the 5' end of our example virus, of our query virus, is very similar to this orange virus here, and this is explained here. So orange is a subtype G sequence, so that means that the 5' end of our virus is most likely derived from subtype G because it shares more than 90% homology to subtype G virus. In contrast, the similarity to other subtypes in that region of the genome is much lower. So that's why this similarity value is below 85% here. But then what you can also see is that suddenly there seems to be a breakpoint, and that here, starting from here in the genome, the red virus is more similar to our virus, and red refers to subtype A1. And then there is even more breakpoints that have been identified by the sliding window analysis. So the bottom line or the conclusion here would be that the virus that we put in, the virus sequence, is not a pure clade, but really a recombinant that is derived from this clade G and A1, and also fragments of the purple one, which would be J. So there have been multiple recombination events that resulted in this new chimeric virus. We also, apart from recombination, we already talked about reassortment, and the prime example that we had already mentioned is influenza virus. So in influenza virus, we distinguish between antigenic drift and antigenic shift. So drift refers to antigenic changes that are the result of single point mutations. So the surface antigens like hemagglutinin or neuraminidase continue to evolve, so they acquire, assemble different point mutations that may change the phenotypic properties of the virus. But these are usually just smaller mutations that usually don't have a large impact on the phenotype. They may have, but usually they don't. In contrast, antigenic shift is caused by reassortment. So if one individual is infected by two different strains of influenza virus, these may reassort their genomes, so different genome fragments may be sorted in a new manner. And this, of course, as you can imagine, may have major effects on the antigenic composition of the virus, and in the end also on its pathogenicity and infectivity. And what you need to keep in mind is that while recombination can essentially occur in any virus, a segmented genome is a prerequisite for reassortment. So only if there's at least two segments, reassortment can occur. And apart from influenza viruses, which have 6 to 8 segments, reassortment may, for example, also occur in Lassa viruses or Hunter viruses that have 2 or 3 segments, respectively. When we did this interactive sorting of the viruses, you also nicely mentioned these examples. These viruses as examples for viruses that benefit from global warming and climate change, because all of them are transmitted via mosquitoes. And there are different mosquito vectors for different viruses. And what several countries try to do is to monitor the spread of mosquito species in different regions. So Europe is also doing this. So this one here shows the distribution of an Asian mosquito in Europe, and we can trace or track its spread in Europe. And the presence of this or specific mosquito vectors will, of course, also be a risk factor for further spread or appearance of the respective viruses. And in this context, I would also like to introduce a few important terms. So one term is arboviruses. Arbo is short for arthropod-borne viruses. So this would be any virus that is transmitted by arthropods. So it's not a taxonomic term like flavivirus or retrovirus, but it just describes lots of different unrelated viruses that are transmitted by arthropods. And arthropods include not only mosquitoes, but also ticks, bedbugs, fleas, for example. And another important term is vector competence. So that means that not every vector is competent to transmit every virus. So that means not all viruses can be transmitted by ticks or mosquitoes, because the viruses also have to adapt in their vector species, to replicate in their vector species in order to be efficiently transmitted. And we refer to this as vector competence. So this allows us to really monitor specific vector species to better predict the risk of viral spread. And one example, as I mentioned already, is chikungunya virus, which is actually or has been spreading a lot in the recent months. So this is again a disease outbreak news article. So the WHO says that in 2025 there was a substantial increase in case numbers of chikungunya virus in lots of countries, reaching a total of almost 500,000 cases until September this year, including several deaths from about 40 countries. And they also propose to enhance vector surveillance and control. And one way of monitoring vectors is actually a citizen science project. So this is an example from Germany where we have the so-called mosquito atlas. And this is a citizen science project where citizens, so non-scientists, just laypersons, are asked to collect mosquitoes in their home, in their bedroom, and to freeze them and then to send them to specific research institutions in Germany. And then they will be analyzed. So the species will be determined. And in the end, this allows us to draw a mosquito map, like a prevalence map, all over Germany, and to more easily detect also emerging mosquito species. And this leads to some interesting findings. So there is even a paper on this. So a few years ago, someone sent mosquitoes via the Citizen Science Project to these research institutions, and then they were identified as Aedes aegypti, which is an important vector for different viruses, including Zika, dengue, and so on, which was not prevalent in Germany by then. So everyone was very alarmed and then went there, and then they set up traps and tried to see whether there's actually more Aedes aegypti species in that area. But in the end, it turned out that the person who sent these mosquitoes in was traveling to Mauritius and visiting relatives there. And when she returned to Germany, she took some plant cuttings with her and put them in a glass of water. And then somehow these plant cuttings contained mosquito eggs. So essentially, she was breeding or importing eggs of Aedes aegypti and breeding them in her home. So they hatched, and that's why they ended up in Germany. So that's just to illustrate that the monitoring works, and that also nicely illustrates how vectors can be transmitted to different geographic regions. Coming to the example of lack of immunity, you also mentioned measles as a prime example. And unfortunately, measles are spreading again in different countries, including different countries in Europe, but also the US. And this is again a WHO report from spring this year, where it says that in the year 2000, measles virus was actually already declared to be extinct or eliminated in the US. But unfortunately, this year, so 25 years later, the situation has worsened again because of vaccination gaps. And there has been a larger outbreak of measles viruses in 17 of 50 US states because of poor vaccination rates. And measles can be a deadly virus and causes severe immunodeficiency, but it's unfortunately frequently underestimated. So we can refer to this as a re-emerging virus in the US. Middle East respiratory syndrome virus is a nice example for a virus that benefits from immune deficiency. So in our interactive slide, we mentioned HHV8 as an example, but Middle East respiratory virus is also interesting because this virus is being transmitted from camels to humans, but it's not very well adapted to humans. So usually there's no human-to-human transmission, or it's very inefficient. So the virus is not very well adapted to humans yet, but it may benefit from humans that are immunocompromised. And when you read these WHO outbreak news, usually the cases that they describe are cases with comorbidities. So many of these patients suffer from other diseases, from cancer or some other kind of immunodeficiency that allow this Middle East respiratory syndrome coronavirus to replicate in these patients. And unfortunately, this virus may or will further evolve in these patients, adapt to the human host, and one could see these immunocompromised individuals like as breeding vessels or evolution vessels that may actually, in the end, allow the virus to better adapt to humans and to result in the emergence of a MERS virus that can be transmitted more efficiently from humans to humans. we also briefly talked about bioterror and bioerror, and you also mentioned smallpox virus. So indeed, smallpox virus has already been used as a bioterror agent. So actually centuries of years ago during the French-Indian War in North America. So there were parties of this war that actually tried to distribute contaminated blankets to the other side in order to spread smallpox among their enemies. And there has also been incidents of unintentional release of smallpox virus at the end of the 1970s. So there was a lab in Birmingham in the UK that, of course, still had smallpox samples, patient diagnostic samples. And then for some reason, a medical photographer got infected. So the exact route of transmission is not known. The lucky thing was that she really stayed at home and did not spread the virus further. But this was one example of bioerror. And I think I mentioned this before, smallpox viruses are extinct in the wild, but there are two labs in the US and Russia that still keep some smallpox viruses in their freezers, arguing that we may need them in future for research purposes. I also mentioned that you as researchers should always keep in mind that it's not only new viruses that may emerge, but there could be entirely new pathogen groups. So we talked about these groups of pathogens before, but there are actually more examples. So there are two very interesting examples. So there are infectious algae. So there's one species of algae that can cause disease, like a dermatitis in humans. And then there's even something that we call infectious cancer. So you may potentially or probably know that certain viruses are oncogenic, can cause cancer, like papillomaviruses, for example, can cause cervical cancer. But the examples that I would like to introduce here are totally unrelated to viral infections. So in this case, it's really the cancer cell itself that is transmitted from one individual to another. This does not occur in humans so far, so we don't know any real examples from humans, but we know examples of transmissible cancer from animals. And actually, the two best described examples can be found in the Tasmanian devil and the dog. And the two diseases caused by infectious cancer in these species are DFTD and CTVD. So what are they? So these Tasmanian devils suffer from the so-called devil facial tumor disease, which you can see here. Essentially, that's a transmissible tumor. So when these Tasmanian devils get involved in fights, they may bite each other. And then some of these tumor cells from an infected individual may be transmitted to an uninfected individual. And surprisingly, they are not rejected, but the tumor just continues to grow in the other individual. So if you sequence these tumor cells, they are different from the infected host. So it's not, these are not transformed cells of the host, but these are ancient tumor cells that have been transmitted for hundreds of years. And for the dog, this is a very similar situation. So here it's the canine transmittable venereal tumor. So this is transmitted sexually, so it affects the sexual organs, the genitals, and during sexual intercourse or mating, these tumor cells can be transmitted from one individual to another. And they have also been sequenced, and they're most likely derived from some ancient wolf-like dog breed, but still being transmitted in the dog population. Again, this is not an example of a virus that emerges, but it's just to illustrate that there might be more than we currently know. So just keep your eyes open and always keep in mind that new pathogen groups may emerge. And for the last category, the special practices or risks, I also picked one from the the WHO. This is actually from 2023, so already 2 years old, but I felt this is interesting. This actually describes the outbreak of botulinum intoxication, again, not a virus, but a bacterial toxin as a result of medical procedures. So obviously a lot of people from Central Europe were traveling to Turkey to get injections of botulinum neurotoxin, but in quite a few cases, these were contaminated, so that it actually caused severe botulism in these patients once they returned to Central Europe. So this would be a prime example of a very special practice or risk behavior. And with this, I'm actually already at my last slide, or second to last slide. I have one final question to you that's more like a general opinion question. So after everything we have discussed today and everything you already know, what do you think? So which virus or which virus family actually poses the largest risk? So if you were a researcher or pandemic researcher, which virus would you focus on? What do you think, which family harbors the greatest risk and would most likely cause the next pandemic? Again, this is like the word cloud that we had before. You can use your QR code scanner and then just submit a reply. And I'm just curious to see what you will answer. I will also add an answer here. So I just give you a minute to reply and then we check your answers. So just keep in mind what we have learned. So which viruses mutate fast, which viruses do have a high reproductive rate, which viruses are humans exposed to, and so on. Yes, so there are a few replies already. I just give you a few more seconds. Yes. Just keep on refreshing, but this looks good. I mean, you can also refresh it on your mobile phone, I think. But what I can see already is that a lot of people think it would be respiratory viruses, right? So there are 3 people that put no 4 that put influenza virus. I actually also put influenza virus. I think that's a good candidate for several reasons. So there is a large variety of influenza viruses already, lots of animal viruses. There have been examples of pandemics in the past that it can reassort and recombine. Someone put zoonotic viruses. Of course, I think the biggest risk comes from these viruses, the ones that spill over from animals to humans. A new coronavirus is certainly also an option. So we have seen coronavirus outbreaks in the past, more recent outbreaks like SARS-CoV-2 or MERS coronaviruses. That's good. Some people also put arboviruses like West Nile, dengue, and so on. They're also likely candidates, but here you have to keep in mind that they will probably only spread in certain regions of the world that have the respective, or where the respective vector species is distributed. RNA viruses are more likely to cause an outbreak compared to DNA viruses because they evolve and adapt faster. Smallpox would be an example for a DNA virus, but certainly it's highly contagious and would benefit from vaccination gaps. This looks very good, I would say. Yes. Perfect. And this, I'm actually on my last slide. So this is just a brief recap. So today in the first half, we learned about origins of emerging viruses, including naturally occurring types of mutations, zoonosis, factors that favor their spread, climate change, globalization, vaccination gaps. And in the second half, we discussed a few more examples. And what you should also keep in mind is that not every exposure to a zoonotic virus causes a pandemic. So there are a lot of different scenarios. And with this, I'm at the end. I would like to thank you for your participation and for your ideas, and I'm happy to answer any questions. Thanks so much.
Thank you so much, Daniel. Apparently, they have understood everything, so you can ask. They don't have any questions.
Sorry, state it again. I didn't get it, Charmaine.
They don't have any questions,
Okay,
so apparently they have getted everything.
that's
You can
good.
ask them.
I can ask them. I'm afraid I haven't prepared any questions. I mean, I asked my questions already. Yes, I mean it's I could ask. So some people put arboviruses in my last question. So it would be interesting to know why they think that arboviruses pose such a large risk to the population. Or what was the reason for selecting phyllo arboviruses? Maybe someone can explain.
Anyone want to explain who put the arbovirus?
Or any other virus, yes.
Adil, you can come here. Adil, you can come here. He cannot listen. Okay, we have a participant from Egypt.
Just for climate change.
Now, can you listen?
Say that again.
You will not
I
be
can't.
audible. I think you will not be audible. Adil, you can come here.
No, sorry, I can't hear you via the microphone. I think you have to come to the front or wherever Shamin is. Yeah, she's hiding.
No, I don't want you to see me.
Hello,
Hi, nice to meet you.
nice to meet you too. I'm Adil Gadada from Egypt. I think that the climate change is a basic reason for arbovirus distribution.
Yes, I think that totally makes sense. And that's why so many people continue monitoring mosquitoes and other species. So maybe that's also something that is done in your countries. And I think this is really important. It doesn't necessarily have to be viruses that are entirely new, but some viruses that we already know may simply spread to new areas of the world where they haven't been before. Yes, so in that sense, I think arboviruses are a major threat. Yes. Good point.
Thank you.
So there are a couple of questions now.
Perfect.
So Rashid will come.
Assalamu alaikum, sir. My name is Muhammad Rashid. I have a question. A person have a virus, but who is the asymptomatic? How we will eliminate the person?
How you mean how to identify the person that
Yeah.
carries the virus? And oh, that's a tough question. I mean, so there's if there's an outbreak, there are of course different screening approaches. So when there was SARS-CoV-1, for example, there have been these temperature screenings, fever screenings at airports to pick out those candidates that show symptoms, but they would not have captured asymptomatic individuals. And that's very, very difficult. Essentially, you would have to take a sample, a blood sample, whatever, and screen a lot of people. So I think I don't have a good solution for this. I mean, you can identify contact persons at risk, put them in quarantine, screen them. But in many cases, it will not be feasible to screen the entire population or all the travelers because diagnostics will also take some time. And that was actually a big advantage during the first outbreak of SARS-CoV-1 because individuals were only infections after they developed symptoms. So that means one could take out or quarantine all the people that showed symptoms before they could infect others. For SARS-CoV-2, this was different because some people were infectious before they developed symptoms. And that might have been one of the reasons why SARS-CoV-1 could be contained, restricted, whereas SARS-CoV-2 spread so efficiently. Good question, but difficult to answer.
Thank you, sir.
So I'm Amirat Lainka from Benin. So I wanted to give you the answer about your question. You ask some of us choose abrovirus because we know today we have increasing in global travel and also global warming that's change in the vector that's lead to the change in the vector distribution.
Yes, valid point, valid answer. It's both. It's the climate change, the traffic, and this example that I mentioned of the citizen science project also nicely illustrates this. So there was this lady traveling from Mauritius to Germany, and she was just bringing these mosquito eggs with her and essentially helping the mosquito to reach new geographic areas. Yes, you're totally right. I agree. Thank you.
Assalamu alaikum. Hello, sir. Can you hear me?
I can hear you. Hello, nice to meet you.
Nice to meet you too. Actually, I have a question. And as I know, Nipah virus has a high mortality rate, 40 to 70%, and we don't have any vaccine yet. So what is the reason?
The reason for not having a Nipah virus vaccine, that's a good question. I'm not a Hendra Nipah virus expert. So the problem is, so one reason could be that there's not enough economical interest yet. So this sounds very sad. So there have been regular outbreaks, but usually it's not super large outbreaks. And usually, unfortunately, bigger companies are only interested if there's a big market for this. I would say that Nipah virus, it is a relevant pathogen. There are regular outbreaks. If I remember correctly, isn't there a vaccine for animals for either Hendra or Nipah? But I have to admit, I don't know very well about these two viruses. Maybe someone else does know more. I don't think it's actually because a vaccine would be so difficult to develop. So my gut feeling would be it's more because of economic reasons, unfortunately. But maybe someone else can comment on this. Sorry. So you should develop one, yeah.
Thank you.
So there's one more. Two more.
This one. This
You just speak.
Okay. Hello. Can
Hello.
you hear me?
Yes.
The question that I wanted to ask you is, how can we classify if this virus is originated because of bioterror or if it is just a normal outbreak?
Tough question. I think, I mean, this discussion is also still ongoing in the case of SARS-CoV-2. So there are quite a few hypotheses regarding the true origin of SARS-CoV-2. Some people say it's a natural zoonotic outbreak. There are some researchers saying that it might have been a lab leak accident. There are a very low number of people think that it was bioterror. In some cases, it's probably impossible to tell if you cannot really trace it back to a very specific accident. But sometimes the genomic architecture of the virus can give some hints. So, I mean, if some researchers would intentionally modify a virus, that would probably leave traces in its genome. Let's say they would combine fragments from different viruses that are only distantly related. We would probably still see these traces of manipulation in the genome because some combinations of viruses would not naturally occur. But on the other hand, there is also natural recombination going on between two closely related viruses. So if we see evidence for chimeric constructs, it's sometimes difficult to tell whether these occurred naturally or in the lab. I guess in many cases it would be impossible to tell.
Okay, thank you so much.
Hello, good afternoon.
Good afternoon.
My question is, at the beginning of the lecture, you were introduced as someone who has some experience with HTLV. I would like to know what your perspective or what your idea would be, the potential of HTLV to cause a pandemic or an outbreak, and the likelihoodness of it to happen, especially in the low-income countries where we do not do testing for HTLV when it comes to the blood banks and so on and so forth.
So I'm actually not an expert on HTLV, so we work a lot on HIV, another retrovirus. So I don't think HTLV would be a major risk. So HTLV is limited to certain geographic areas. So some types of HTLV can be found in Japan, but there's not a lot of, so it's not very infectious, so it doesn't travel around the world, let's say. So I would say the risk for HTLV causing a pandemic is not that high.
Thank you. Thank you so much, Daniel, for informative lecture and the active participation.
You're welcome. I have to thank you for your participation. Thanks so much.
And we hope to see you soon again in Karachi.
Same.
Have a good
Okay. Have a good time. Enjoy the rest of your workshop.
Okay.
Thanks again. Bye-bye.
Bye.